So the last speaker in this session is Dr. Mark Gardner, who's a computational and medicinal chemist. After a first degree in chemistry from Imperial College in London, he moved on to natural product chemistry and computational chemistry at Cambridge before joining Pfizer. During 19 years at Pfizer, Mark set up and managed the lead discovery group, ran a file enrichment program with a huge budget of around $90 million, and introduced a number of important new design tools. In the last couple of years, Mark's been working on a variety of global health projects, malaria, TB, schistosomiasis, leash, Chagas, lots of our favourites, with a number of organisations. And it's the subject of his talk today will be looking at computational methods in neglected diseases, drug discovery. So Mark.
Thanks very much, Charlie. Thanks for the introduction, and thanks very much for the invitation to speak here. With tongue-in-cheek, I should thank Thierry as well for reminding me and the other sandwich Pfizerites of the closure of the site. I guess I have some good news to report. There is a phoenix rising from the ashes, and the new owners tell me there's about 1,700 people going to be employed on the site by the end of the year, which is good. 600 of them actually employed by Pfizer, or thereabouts. So it wasn't a complete closure in the end. They did close the discovery part. Anyway, one of the organisations, or even two of the organisations, I guess, that are on the site are represented here, and I guess I represent both of them. On the left-hand side, I'm doing consultancy in neglected diseases as AMG, and Solvensis is a not-for-profit, a very small not-for-profit. We have a collaboration with Quentin Bickel at the London School of Hygiene and Tropical Medicine on a schistosomiasis drug discovery project funded by the MRC. And they've got a really nice screen, actually, and we promised to screen 200,000 compounds, and that's going really well. In fact, they're working their way so fast through the set of compounds. If anybody's got a nice set of 50,000 or so compounds, I'd love to talk to you. That would be great. Anyway, what I'm going to talk about today is two things, and they very much sort of relate to the topics that Jean-Louis Raymond and Thierry have been talking about, and it really relates to all about the importance of finding good leads. And I'm going to do two topics in that space. One, looking at just what happens when you screen large file decks and what factors affect hit rate, and are the differences between phenotypic screens for neglected diseases and biochemical screens in human disease. That's work essentially analysing Pfizer HDS data. The neglected disease stuff is in collaboration with MMV and DNDi. And then the second subject, looking at using information from HDS to find new leads. And as I guess I'm the last speaker, although there's a panel session afterwards, I guess I'm the last speaker before the drink session. My link to that is that we're going to look at the similarity between computational chemistry and wine tasting in that section. So, keep alert for that. Now, the first thing I'm going to talk about is, why is it so important? Why are we talking about finding hits? And why is that so important? Obviously, you've got to have a starting point. That's clear. But I guess the other thing about hits is that it turns out that talented though medicinal chemistry is as a discipline, and medicinal chemists are as individuals, the plain truth is that the candidate molecule often looks quite a lot like the hit that you start with. And obviously, if you don't start with a decent-looking hit, you're going to have your work cut out to get yourself a decent-looking candidate. On this graph are shown pairs of compounds, which are so each dot represents a pair of molecules, the hit and the candidate, or the lead and the candidate. And it's plotted in 2 dimensions because I'm just looking at 2 different ways of measuring chemical similarity. What you need to know is that towards the top right, these are really, really very similar pairs. So I've shown some examples of those that are more in the bulk here of pairs of molecules that are actually a a bit more different, at least as this graph goes. But you can see even, looking right down in the bottom left, you can see the family resemblance of the candidate to the lead, and that's true, I guess, for these others. And I think most of you that have, been aware around medicinal chemistry and seen stories like this will know what I'm talking about. You've really got to start with a decent lead if you want a good chance of getting a decent candidate. And I could have put up any one of a number of publications on oral drug properties. This is a relatively recent one that Paul Leeson's done, where he was also looking at different companies and their approach to drug-like properties. But basically, the message is that, at least from a statistical perspective, you can have some handle on the kinds of compounds that are likely to give you good oral drugs. It's not to say that you can't get good oral drugs from other compounds as well, but from a statistical perspective, all these analyses point in broadly the same direction. And given that in neglected diseases we're talking about, for the most part, with like once a day or maybe single dose cures, simple formulations, maybe mass drug administration. Probably these requirements are probably at least as true in neglected diseases as they are anywhere else. So I'm going to move into a sort of analysis of the properties of hits in high throughput screening now. And as a sort of, the reason I got into this really was analysing what Pfizer had done in this file enrichment project. As Charlie said, I was responsible for one of the collaborations. Depending on who you talk to, I was either responsible for burning $90 million bills or actually did something productive for the company. But anyway, be that as it may, the sort of the genesis of this file enrichment program, I guess, came out of, I think, really a pretty bold leap of faith, maybe a bold leap of faith that you could take at the time when Pfizer had plenty of money. The thought process went pretty much along these lines, I think, and this is from a publication from George Milne. And it was really basically that, if you would assume that you've got a druggable target, then somebody somewhere is going to succeed in getting a drug to the market for that target, assuming that the biology all stacks up. Big assumption, but anyway, if it's a druggable target, somebody's going to get there. And the rationale was, well, at the time, I think Pfizer's file was around 1 million compounds or so. Pfizer is not always the one getting the drug to market. The rest of the industry at the time, I think George had estimated somehow at about 3 million. So the rationale was, well, if Pfizer had a file that was 3 million, then we could get to the market first for that target more often. And what we actually did then was, through 4 collaborations, essentially go about collecting an extra 3 million compounds or thereabouts. And so the analysis I'm going to show is just effectively splitting the compound collection in high throughput screening into 2 sets. The file enrichment set and the non-file enrichment set, which includes, for the most part, a lot of singletons that have been made over, a large number of years in medicinal chemistry, animal health chemistry. It includes compounds, acquisitions from mostly of singletons over the years from a variety of sources, as well as, Warner-Lambert and Park-Davis compounds as well. And in terms of the file enrichment compounds, obviously they were designed with the rule of 5 in mind, and lo and behold, the distributions are pretty much where you would sort of expect them to be. You might argue that there's some peaks towards the limits of the rule of 5, the upper limits, I guess that's partly with applying sort of cutoffs in the design of the compounds, but predominantly, they sort of fit where we intended them to fit. In terms of impact on Pfizer drug discovery projects, this analysis is quite old. These I'm just showing you some of the candidates, the patents that have been filed on the candidates. So at the time there were around about 8 candidates where the hit molecule originated from a file enrichment compound. Obviously, you'll be aware that it takes time for compounds to get into the file, to get screened, for the optimization process to happen. So what the number is now, I couldn't say. I don't know. But back then, it was around 8. And then, of course, there were the projects where optimization had used some of this technology. So parallel synthesis optimization technology and that kind of stuff. And obviously, there's quite a few of them. But in terms of their performance in HDS, you've already seen a little bit of these kinds of plots. I'm going to talk about looking at how the file enrichment compounds and how the legacy compounds compared in file enrichment in high throughput screening. In Pfizer at the time, most of the high throughput screening that was done on essentially all the file or very substantial portions of it was done in these kind of 3 phases. So the first one is sometimes actually in mixtures of compounds where you're just doing single concentration, potentially in mixtures, which it's a bit complicated, but there's a sort of self-deconvoluting idea, so it's not a question of having to screen all of the compounds in the mixture. You're looking for 2 wells to light up that contain the same compound. Anyway, you then do a confirmation of those things that you think might be active, and you can see in that plot too, that a large proportion of the things that you thought were active in the first place turn out to be false positives, and some of them are genuinely active, and so you get a sort of confirmation rate at that point, and then you go on to do a dose response. And in this case, this example, you can see that the majority of compounds that looked active at 10 micromolar do give you a dose response value if you go up to sort of 30 micromolar top concentration. So the reason I got into doing this analysis is really summarized on this graph. So if you look at on this slide in the table, if you look at a large number of high throughput screens, the plain fact is that the file enrichment compounds have a lower hit rate than the legacy compounds. Okay. Now, when you just spent a rather large amount of money, doing the file enrichment, somebody somewhere is going to want to know why is the hit rate low. Not trivial to answer. So, done some analysis which I'm going to talk you through in brief. So, the biggest difference, it turns out, is in that confirmation rate phase. So I've used that as a sort of shorthand for the analysis. But you can see in the overall cumulative effect of the 3 stages, the file enrichment compounds on average are about 3 to 4-fold less likely to have a confirmed dose response activity than the non-file enrichment compounds. So first of all, just looking at some of the properties of the compounds against this confirmation rate. So the file enrichment compounds are in red in these graphs, and the non-file enrichment compounds are in blue. Obviously, you can see a significant difference in the confirmation rate, and you can also see a trend with molecular weight. I don't think many of you would be as surprised to see that trend, and you probably wouldn't be surprised to see a similar one with lipophilicity. Perhaps less marked in the non-file enrichment compounds than the file enrichment ones here, but still, clear. The charge state doesn't make all that much difference. The size of the circles here represent the numbers, so you can see that there's very few Zwits and not very many acids compared to the others. Aromaticity turns out to make probably the biggest difference in terms of certainly looking at the file enrichment compounds and the confirmation rate here. So you can measure it by looking at the number of bonds which are aromatic, or just the number of atoms which are sp2. Obviously that's not just aromaticity, but it's probably a fairly similar property you're measuring there. Obviously some of these properties come with their burdens. In fact, most of the ones that correlate with activity usually correlate with some problems as well. And this is just, I could have shown similar kinds of things for lipophilicity. This is just looking at aromaticity. On the left-hand side, this is in-house data from Pfizer, which is just looking at a particular toxicity assay and showing that there is a greater degree of toxicity for compounds that have a lot of aromaticity. On the right-hand side is a publication talking about the impact of aromatic ring count on developability. So same message coming through from the two different analyses. One thing I did do to try to understand to what extent are property differences responsible for this difference between the file enrichment compounds and the legacy compounds was essentially to bundle all the data together, try to extract a calculation which would predict conformation rate for all compounds, and then apply that calculation to all the compounds to see, well, if you just apply that calculation, can you tease out what proportion of this difference between legacy compounds and file enrichment compounds is due to properties. And it turns out that around about half the difference, I would say, that you see in terms of conformation rate is due to this property difference, okay, which then begs the question, well, what's the other half due to? So I'm going to show you some structures. So one of the things I did then was to look at any structural differences between compounds with high conformation rates and low conformation rates. So here's a bunch of ring systems with very high conformation rates. You can see there's a lot of aromaticity here. Not all these ring systems are necessarily particularly desirable. It looks like the kind of thing that might intercalate in DNA or have various other issues, but certainly it correlates with the idea that lots of aromaticity tends to give you high conformation rates and high hit rates. Here's some low conformation rate ring systems, and again, these are ring systems with more than 100 examples in the file, and you can see some of them 1%, 2%, so very, very different from the 50 to 90% conformation that we were seeing before. And of course, these predominantly are not aromatic, or aromatic with non-aromatic parts added to them. I guess what's interesting, and I guess Thierry raised it earlier about rings or compounds in people's file which have been screened against a lot of high throughput screens being inactive in all of those high throughput screens. And I know that I used to have discussions with someone at Pfizer about this who said, well, some of these compounds are never going to be active. Yeah, I don't know. I don't think we can say that. Personally, I like a lot of these ring systems. If one of these came up active, it would be great. And it certainly does contain, or quite a lot of them contain 3D character. It's also true to say that quite a lot of these are in the file enrichment sets. So in some ways, some of the file enrichment work at least was taking what looked like quite interesting rings and elaborating them in parallel synthesis. Well, if your motivation is to take a quite interesting ring system and elaborate it, that's quite different from taking an active compound against a kinase and making lots of analogues of it in a kinase project. And maybe that's part of the reason for the difference between the file enrichment set and the non-file enrichment set as well. That, okay, so properties explain some of the difference, but the legacy compounds were all made really predominantly with a purpose of, and they were close to something that was already active against something. And when tested, sometimes against a related target, maybe it's not surprising that they're going to be active. Some of the file enrichment compounds, at least, were made, because there was quite an interesting ring system, and we'd like to elaborate some of those things. And there was no real certainty of activity. It's true to say we've sometimes made an awful lot of analogues of those things that had interesting-looking ring systems. Talking about having made a lot of compounds that were very similar, this is a presentation, I think, from Jeremy Everett, again at Pfizer, looking at some years after the file enrichment work completed, looking at an analysis of compound diversity in the file, and partly reflecting a concern that high throughput screening with all these compounds, if you screen the lot, is getting very expensive. So part of the goal here was to look at selecting compounds that would be representative of the whole collection and slightly less expensive to screen, but not throwing away the diversity that was present in the file. And I think the key bullet on this slide is the purpose is not to mine out the SAR. So if you're trying to do a high throughput screen where you just want to find a hit, you don't need lots of analogues necessarily. You certainly don't need lots of analogues that are active just in order to find an active hit. So if your goal is just to find the hit, you don't necessarily need loads of very similar representatives. And that was essentially the mindset here. And the way that this work was done essentially was to use this ECFP6 and a Tanimoto cutoff of 0.6. It's quite similar to what Thierry showed. And they decided that they wanted no more than, for each compound, for it to have no more than 40 analogues that were that similar to it within the file, and to remove compounds from densely populated clusters, basically. So what's the impact on HDS output? This is obviously a simulation, what would happen in screens that have already been run. So the green bars here are showing the number of hit series in various different target high throughput screens, and the small red tops to these bars are showing the number of active series that would be removed having done this analysis. Obviously, if your compound is quite unusual and there aren't neighbours to it, it wasn't removed. Okay, it was only depleting the numbers of very similar compounds. So that was just looking at the fine enrichment compounds, comparing them with the legacy set, and across a range of mostly biochemical assays. This one is now comparing those biochemical assays predominantly with phenotypic parasite HDSs. So on the right-hand side, we're looking at falciparum, leishmania, T. cruzi, T. brucei, and they were done in collaboration with MMV and DNDi. And then on the left-hand side, we're looking at essentially the set that I just showed you. So really, I guess what we're asking here is, is there something different about neglected disease, chemical space, at a very crude level, just properties like molecular weight and C-log-P and stuff like that. So you can see again there's an increasing trend with molecular weight. In the legacy set, it turns out there's quite a lot of macrolides, and those show up with quite a lot of activity in some of these screens. Lipophilicity, again I'm showing an increasing trend. If anything, probably this is more marked with these screens than with the predominantly biochemical assays, but it's still obviously very much in the same direction. And with the aromatic bonds, again, greater aromaticity leads to higher hit rate, pretty much the same as we saw in the biochemical assays. So essentially, comparing the two, I guess my crude analysis would be, there's kind of no magic chemical space that is specific to or of most interest to neglected disease chemical space. It's really it's the same in terms of phys-chem properties as it is for, and the rules are the same as they are for biochemical assays at a fundamental level. In terms of acknowledgements for this part, well, obviously in terms of the fine enrichment, it was a very large number of people who devoted a large part of their careers to it. I've also noted some of the key movers in Pfizer of that initiative down here, and DNDi and MMV with the screening and the NTD part there. So I'm going to move on to the second part, talking about finding leads for leash, Chagas and hat diseases, and we're looking here at doing some virtual screening, and also looking at can we do some virtual screening using some proprietary information without revealing the structures behind the proprietary data. So it's important, obviously. I think we've heard all afternoon about some of the work that's been done to put screens together and to carry out screening. Some of these diseases, I guess leash in particular, hit rates are really pretty low, and finding good leads is very difficult. So we do need to make the most of the few hits that we've got. And one thing that we can do is to take some of the activity that we do have and to build some models and see if we can point those at commercial collections and find some additional compounds. And that's some work that's ongoing. I'll touch on that. And also to use these same models. DNDi are talking to some of their partners to be able to use those models with some of their pharma partners as well, which would be fantastic. So in terms of the methodology, this is thinking about doing the virtual screening. So to those of you that are computational chemists, you've already heard some of the methodology explained here. To those of you that aren't, I guess I think in a sort of way, maybe stretching a point, an analogy for me is we're taking these active structures and trying to build some memory or image of the active compounds. And it is a bit like, I think, someone who's good at wine tasting, thinking about, well, this is an oaked wine, white wine, and if I taste another one, I'll remember some essence of that, and so when I use that knowledge against a whole bunch of other wines, I can detect the fact that it's another oaked example. So using this memory or this image, the model, in screening a large compound database, we're essentially looking for echoes of the training compounds, echoes of the active compounds that went into the model, to pick out a new set of structures that hopefully will have a higher hit rate than just a random set of compounds would. Okay, now in this particular case, so I'm looking at just before we get on to that, I'll talk about the objectives for the ligand-based virtual screening. So really hoping to find analogues of existing series, I guess particularly those where they might be a little bit more different from the actives than you would maybe synthesize. So, once you start walking away from active compounds, you don't have to walk away too far before you quickly make lots of inactives, which is obviously a bad thing to do. You can afford maybe to have a slightly lower hit rate if you're just picking compounds that already exist. Also to mix and match structural features from different active series. I mean, that's something that medicinal chemists do in synthesizing compounds, and again, useful to be able to do that from the file. I won't talk about the method that was also applied here, but the other thing that was looked at was picking compounds, displaying 3D features of the different actives as well. So what I'm going to talk about is a method that I should have credited Evotech here, who did this work, predominantly building a Bayesian model using NIME, which is, if you like, as a shorthand, you could argue it's a pipelining tool, a little bit like Pipeline Pilot, but it's freely available, and also Morgan fingerprints. And we'll come on to this point about trying to do that in a way that doesn't reveal the structures of the training set. So here are a couple of graphs showing QSAR models, actually showing recovery experiments using QSAR models, and it's comparing the model that you can get using the data that was available on T. brucei with some proprietary data from pharma companies, and comparing it with the model that you could get were we only using non-proprietary data. So on the right-hand side, we're only using the non-proprietary data, and what I've done here is we've compared the actual model, which is the red line, with a model that you could generate if you scramble the Y values in the training set. So essentially what that's doing is taking the compounds and randomly assigning them to activity data. Okay, so what you would hope is, obviously, that your real model is much better and quite distinct from a model where all you've done is essentially thrown some random numbers into the pot. On the right-hand side, you can see that unfortunately that's not the case. So you want a line that is pretty much, well, like the red one on the left, basically, or even closer to the top left-hand corner, which is showing in this rock plot, for those of you who know about these things, a good discrimination and a good ability to predict activity. On the right-hand side, without the pharma data, essentially this model is pretty worthless. It's just showing that it's essentially overtrained. On the left-hand side, including the proprietary data, you get really good separation between the real model and the Y scrambled data model. So essentially that was done using these Morgan fingerprints, and essentially that's just a simple way to think about it is taking the model and taking a molecule and breaking it into its component parts, and then assigning some a presence or absence of these features in a bit string. The translation between features and bit string is a bit complicated, but that's in essence what you're doing. And then you assign a score to those bit strings. So here I'm showing an example where there's a feature which has got a high score, and it's got a high score of 3.3 because in this training set there are 36 active molecules with this feature in it and 9 inactives, and in high throughput screening that's a really high ratio of actives, right? In the lower compound this feature has got a low score, a negative score, because all 916 examples with this feature in it were inactive, okay? And effectively that's how the model's working. It's just chopping up your test compound and assigning scores to the different features based on what was in the training set. So here's the model with a list of features and a list of scores associated with it. So as I mentioned, one of the things we want to be able to do is to use these models in situations where it's important that we don't reveal the exact nature of the training set. So we want to understand what can you tell from using one of these models to when you screen a compound set. So this is an example where I'm only using a model that was generated on non-proprietary data. The example I showed you before, that was a lousy model because it was, I think it was T. brucei. This one, T. krusei, is not bad actually because it was a much larger non-proprietary data set that went into building this particular model. So that's the one that we're using here. And this just shows effectively that the training set compounds in blue score about the same as a test set, and artificially we separated a bunch of compounds that were really from the same series, half of them into the training set and half of them into the test set. You can see that in terms of score there's no real difference between the test set and the training set, so you can't differentiate, you can't identify just by looking at the score which one of these compounds was in the training set and which one was in the test set. We also did some tinkering about with the scaffold and also the compounds that have had the scaffold modified a bit as well. You can't really differentiate which was the original scaffold and which was ones that we've tinkered about with. So in conclusion for this part of the talk, basically I guess the key thing is that where pharma companies have let DNDi and us use their data in building models, that's extremely valuable because without that in many cases we get models that really aren't workable. The models themselves are blinded to the training set, and then in terms of using the model, it obviously finds, you will have seen if you're close enough to the front to see the structures, it obviously finds molecules that are very similar to those that are in the training set, but you can't tell whether those compounds were in the training set or not. And obviously screening data from this work is the key thing we're waiting for, which I would love to have been able to share with you, but timing is not done yet, I'm afraid. So just for this second part, acknowledge Alex who worked with me on this, and the guys at DNDi and Evotech, and also importantly the pharma folks who let us use their data. And then my very last slide is I guess just some personal sort of perspectives on thinking about selecting compounds for HTSs for NTD. And I guess from the first part of the talk, really just saying there's no, unfortunately, I think, no magic property combinations in chemical space for NTD. I think probably we kind of all knew that really, but I think it's true. We just have to live with it. And really, these activity versus the sort of ADME and tox battles that everybody's familiar with, it's really the same as in other human drug targets, more or less, with some wrinkles. And also to remember that the clinical candidate will usually resemble the screening hit. So it is important that we find good hits. To which end, obviously, as we've been talking about, it's very important, as people have been saying, that we screen in assays that translate. And if we've got a choice, and especially if we haven't proved that chemical space that's relatively translatable won't work, that's a great place to start. Obviously, if we're starting to find that we're not getting many hits from that, we may have to look elsewhere. I think it is worth incorporating learning from what's worked before in terms of activity models and things like that. But novelty, especially if you can convince yourself that the path through development isn't going to be complete torture, is definitely going to be worth doing, especially when you've looked for the low-hanging fruit. And also that certainly access to pharma companies' compounds and data and information about those compounds can be enormously enabling. Thank you.
Mark, thank you for giving us an insight into industrial drug discovery and some of the lessons from 10 or 20 years of high throughput screening and compound collections and file enrichment, and also how some of these new tools are getting applied to NTD research today. So thank you very much. Questions?
Question in terms of creating Bayesian models for phenotypic screening against a whole organism. Other than T. cruzi, where most of the compounds hit the same target, CYP51, I don't see why a Bayesian model would actually work, because you're looking to hit multiple targets, so you're creating a whole mismatch of models to hit multiple targets.
Yeah, I think it's an excellent point. I think that one of the features of the way that the Bayesian model works is it's not, it copes with that fact that, yes, there are multiple targets in there and multiple series that hit multiple targets. It's not going to be perfect, and the sort of mix and match idea, that would probably more rely on the idea that you're hitting the same target with the same series, or not the same series, but hitting the same target at least. But certainly the recall that you can see with that kind of approach, I think it suggests that it can work. I think you do have to be very careful. You're right, with phenotypic screening and building models. I think you can't go building or trying to overlap different series and thinking about how do they overlap, for example, in a 3D sense, when you've got no idea that they hit the same target, for example. So I think your point is well made, that you do have to be very careful about model building in those situations, and it's less good than if you know the target. I think that's true, but I still think that the Bayesian model can find useful things in that context.
These are terrific talks, and the message that comes through to me is unambiguously this is an argument for open innovation in the chemistry world and pharma. It's overwhelming. And the reason is obvious both in all three talks, but particularly in Terry's. Earlier drug discovery, every company's prisoner of its past, and so the genealogy of the chemical base, the database you have, is based on what you've discovered in the past as safe and effective drugs. So what you've taken are millions of years of evolution in biology, and you've actually crafted chemical space around that to both make something that works at the target for efficacy, and also, in many cases, has distributed elements in there that also avoid toxicities of many different types, even to the level of animal toxicity. So that embedded in that information is so much that as smart as medicinal chemists are, they don't understand all of that over the way 3 million years of evolution understands it. So every company then has its experience with biologic evolution and smart chemistry, which was very hard to get to. And so what we're talking about is, can chemists figure out enough to replicate that in 3 years when it actually took a much longer time to develop those different databases? And then the examples that you gave, Terry, unambiguously show that you can prosper tremendously in a quite rapid way just by getting one other company's database. So if you could pick the companies with the most diverse histories of drug discovery and have them all contribute their compounds to a database, it would help everyone else in the industry as well as help those of us on the neglected tropical diseases or any of these chemistry efforts in a way that all the rest of this wouldn't come close to, because look how little time it took to come up with that evidence compared to the complexity and difficulty of doing medicinal chemistry. I would say, I know you published that paper, but this has got to shout out to CEOs of companies that there's a better way of doing it. It's ridiculous to take this many years to discover drugs, especially when people need drugs. I mean, there are many unsolved illnesses, let alone the ones we're talking about. And to waste time with this stupid competitiveness on chemistry when you can come so far with a little collaboration just seems like it's the modern insanity that's burying the pharma industry.
I don't think it's a question, but my answer is yes. Definitely.
As a former employee of a company in that business, it's just painful to see it happening.
Yeah, I mean, I think things are moving in that direction. Maybe not as fast as we'd like to see. Yeah.
Alan.
Do you think we've been too prescriptive in how we've selected compounds for our screening libraries and maybe let our knowledge of medchem override what's in the clinic? I'll give you 2 examples. Miltefosine is a long-chain quaternary ammonium compound, which I bet most chemists would say, well, that won't be orally absorbed and it'll be toxic. And we all know it's being used, first oral drug for leishmaniasis. The second example is, who would have ever thought that an ozonide would have been stable enough to actually go in the clinic? And yet look what's happening for malaria. And I'm just wondering if maybe we should be a bit more lenient in what we put in the screening libraries, but use our medchem knowledge, if we get activity in the primary screens, to immediately test and see if our fears are founded. You know, if a compound looks unstable, if it's active, let's test it in the next screen to see if it actually is stable.
Yeah, I absolutely agree. I think there's some fantastic innovative stuff done looking at, artemisinin and the work there and taking that to OS439 and other examples that you mentioned. Yeah, I think there have been times when we've been too, ready with the medchemist red pen. I guess I'm learning certainly that, we need to be a bit more cautious about that. I still think that there's probably room to say, when we haven't run many screens yet, why not look for the low-hanging fruit? But when it's clear that that isn't giving us what we need, there are situations, obviously, in which other types of compounds that maybe, surprise us a bit, can be excellent compounds. So yeah, overall, I agree with you, basically.
Yeah, I would just follow up on that statement in the sense that we need to remember we are not making the next Lipitor. We're not making a drug that someone will take for the rest of their life, and therefore we have to be really, really careful in terms of its safety. We're talking about infectious diseases where the product profile hopefully would be days. Maybe with Chagas disease it might be weeks or even months, heaven forbid. But we're not talking about years, and that puts a very different tenure onto what you would put in your screening library. You mentioned artemisinin, and I was just about to mention that myself, because a peroxide, that's too reactive. We shouldn't go through that. Penicillin is an irreversible inhibitor. So there are a lot of things that would have fallen by the wayside that are excellent drugs in the clinic, that are lifesavers, really. So I think we just need to be a little more careful.
Yeah, no, I think it's a great point. I guess for the most part we're still talking about oral drugs, but I think on the safety side of things and you know, just looking at things and thinking, oh, it's unusual, therefore I don't like it. Yeah, we probably don't want to be doing too much of that in neglected diseases. I agree.
So maybe last question from Carl before we go into the debate.
I guess most of the phenotypic screens are often run in parallel with a cytotox screen. Did you look at trying to combine some of that data to create a cytotoxicity model? And if you did, was it usable?
So we did do it, yes, in the virtual screening. Yes, we did do it. I'm trying to remember exactly how. I think it was one of the things that was in there, wasn't it? It wasn't sort of a large part of a sort of cut-off, yeah.
But
it was
you
in there
felt
as
you
information.
generated a model that wasn't overtrained, that could discriminate?
I'm trying to remember exactly that.
There's a nice example, actually. It comes back to the question about Bayesian models on a whole cell screen. So it's actually a paper from someone at Pfizer as well, looking at a polynodal cytotoxicity model. So taking data from about 26 different cytotoxicity models and producing a global model that then was quite good at discriminating between toxic and non-toxic compounds. So I echo Mark's comments. I think we may be asking a lot, but we're desperately trying to find some good new series, and so it's worth exploring what the limits of those technologies are. And I think the cytotoxicity model was something we used to inform our choices, but it wasn't a hard cut-off. So again, coming back to the point of being inclusive while we're searching for the initial active. If there's no more immediate questions, I think now is maybe a good time just to pause and move into the panel debate. So I'll invite Rob to come back and moderate us.